At MD Anderson, what we've undertaken is we are using a second generation CAR construct that signals through CD28 and CD3 zeta targeting CD19. What's unique about our product is that it's using a non-viral transduction system. It's a DNA transposon transposase system called Sleeping Beauty, and we have been really leading the efforts to take this into the clinic. And in our first, what we call our first generation CAR, we established the safety and feasibility of this product. And now we really are looking and modifying the trial to assess efficacy, and we've tweaked it some to see if we can improve results. So what I'm presenting at ASH this year is a second, what we call our second generation trial, in which the construct has been revised or modified so that we've switched out our IgG FC domain for CD8, and most importantly, we've shortened the manufacturing time. We don't expand the cells as long ex vivo with the hopes that the cells have less exhaustion. And we've changed the study design so that patients need to have active disease to receive the cells so that we can get a sense of efficacy. And what we're presenting at the meeting this year is results of 8 patients that have been treated to date on this dose escalation study. We've completed our second dose level, so patients have been dosed at a maximum of 1 × 10⁷ CAR T cells per kilogram. And what we're seeing encouragingly is that for the first time we're able to detect the CAR T cells in the peripheral blood, so we can detect the T cells by flow as well as by PCR, and in the first generation trial we could only detect by PCR. We're also seeing responses, albeit transient, and for the first time we're seeing cytokine storm, so we've had a maximum of grade 2 cytokine release. In general, however, the T cells are still really well tolerated, and the majority of patients just had grade 1 CRS. So in terms of the safety profile of this drug, of this construct, it's very safe. In terms of efficacy, we still need to work on the efficacy. We're still dose escalating, so we'll see what happens at the higher dose levels. But it is encouraging that we're able to now detect these cells in the peripheral blood by flow. Ultimately, our goal is to go towards what we call this point of care product, because one of the limitations in all of these CAR constructs is that it takes time to manufacture the product. So until we use an off-the-shelf product, you really need to have a product that you can use on demand. And so our next generation trial will have an IL-15, membrane-bound IL-15 to input into the construct, so that hopefully really all of the culturing or the in vivo expansion, the expansion can occur in vivo. And the idea is we can take a patient, apheresis the patient, they come into the hospital, they get 1 or 2 days of lymphodepletion, and then they get the CAR infused right back. And that's the ultimate goal, and so we hope to open that trial in the spring.
So you're going to be looking to recruit more patients?
This is the current trial is a 4 dose level trial, so we will max at 24 patients, and in the next trial we're going to be looking at 2 dose levels. It's going to be a smaller feasibility trial initially in this point of care, and we'll see what happens.